In this chapter, we are going to learn about amino acids. Let us first get to know about amino acids. It is a compound that contains both the amino group and also the carboxyl functional group. So from the general structure of an alpha amino acid, you can see over here that there is two functional groups present, which is the amino group and also the carboxyl group. Now, what is an alpha amino acid? It is an amino acid in which the amino group is on the carbon adjacent to the carboxyl group. As you can see in this structure, the carboxyl group is given here, COOH. So the carbon which is adjacent to the carboxyl group or next to the carboxyl group is called the alpha carbon. So an alpha amino acid is the amino group which is attached to the carbon atom next to the carboxyl group. So we call it as alpha amino acids. In this table is listed 20 alpha amino acids for your reference. You may get to know these structures of amino acids. Next, we shall proceed with the nomenclature of amino acids. Now, amino acids are named by considering them as an amino derivatives of carboxylic acids. For example, in this structure of an amino acid given, the carboxyl group is numbered as carbon number 1 and followed by the chain C1, carbon number 2, C2 and C3. So the name of this amino acid is 2-aminopropanoic acid. The next example, in this structure, the carboxyl group C, the C is given carbon number 1 and is numbered according to the picture over here. So this structure will be named as 2-amino 3-hydroxypropanoic acid. 2-aminopropanoic acid is also called as alanine. And 2-amino 3-hydroxypropanoic acid is also called as serine. These are the common names of the structures. Let us look at some examples on naming amino acids. How would we name this structure? The carboxyl group takes carbon number 1, this is carbon number 2, and carbon number 3. So at carbon number 2 we have an amino group, and carbon number 3 we have a phenyl group. So this is named as 2-amino 3-phenyl 3 carbons as the parent. So we will name it propanoic acid. Let us look at the next one. There are 2 carboxyl groups over here. So which carboxyl group takes number 1? Yes, the one at the bottom here. This is because when we start counting this carbon as number 1, we have the amino group at carbon number 2. Go on with carbon number 3, 4, and 5. So this will be named as 2-amino pentane dioic acid. What about the third structure? Maybe we can draw this structure out. NH2, CH2, 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 CH, NH2, and COOH. So I have carbon number 1 over here, 2, 3, 4, 5, 6. So this structure will be named as 2,6-diamino hexanoic acid. I hope you understand the naming of amino acids. Let us now look at what is called a zwitter ion. A zwitter ion is a molecule with an overall charge of 0, which has a positive charge on one atom and a negative charge on another atom in the same molecule. The example is given here. This molecule will have an overall charge of 0 because it has a positive charge on one atom here, and also we have the negative charge on another atom in the same molecule. So this is called a zwitter ion. How does a zwitter ion exist? Or why does a zwitter ion exist? An amino acids can undergo an internal acid-base reaction in which a proton is transferred from the carboxyl group to the amino group to form a dipolar ion, which is called the zwitter ion, meaning that there is a transfer, an internal acid-base reaction taking place where a proton is transferred from the carboxyl group to the amino group, which causes the carboxyl group to be negatively charged and the amino group to be positively charged. Next, we are going to look at another term called isoelectric point, PI. Isoelectric point is actually the pH at which an amino acid exists primarily in its neutral dipolar zwitter ion form, and the concentration of the zwitter ion is at maximum. As we know, zwitter ion is electrically neutral and it exists at a specific pH, and this pH is called the isoelectric point. Each amino acid has its specific PI or isoelectric point. Let us take an example over here. glycine. This is the structure of glycine. And glycine's isoelectric point given is 6.1. Now, let us draw the structure of glycine at its isoelectric point, Pi. Now, glycine molecule, which has this structure, has to be drawn at its isoelectric point. So at isoelectric point, the amino acid or glycine exists as a zwitter ion, or we say that the zwitter ion predominates. So the structure of the glycine must be in the form of a zwitter ion. Okay, let us see now. How do we draw the structure of glycine in an acidic medium? Now, glycine in an acidic medium will act as a base where the amino group will gain a proton and glycine will exist as a cation, or we say that a cation predominates. So here the amino acid acts as a base by accepting a proton. In a basic medium, glycine will act as an acid. Glycine will act as an acid where the carboxyl group will release a proton, and so glycine can be drawn like this, where the anion predominates. In order to understand how to draw an amino acid at its isoelectric point in a basic medium and in an acidic medium, let us look at this example. Serine with an isoelectric point of 5.7 has the following structure. This is the structure of serine and its isoelectric point is 5.7. Draw the structures of serine at pH 3, pH 10 and its isoelectric point. Okay, let us draw the structure of serine at pH equals to 3. Now this is a acidic medium. Now serine will act as a base in an acidic medium by accepting a proton. So the structure of serine must be in a cation form, NH3+. The amino group acts as a base by accepting a proton. So serine acts as a base and forms cation. What about at the isoelectric point? The isoelectric point is at pH equals to 5.7. So at isoelectric point, serine exists as a zwitter ion. This is how the zwitter ion of serine looks like. Then at pH equals to 10, which is a basic medium, serine will act as an acid by releasing its proton. So the carboxyl group, the proton will be released by the OH and form an anion.